Microalgae are quite important from an industrial perspective, economically, and also scientifically, because you can harvest pigments, lipids, proteins, can be used in cosmetics for coloring, for nutritional values, so it could be helpful to replace meat protein. Additionally, people are trying to cultivate them for some sustainability practices. So one of these practices is to have a large pool of microalgae, and essentially what they do is they uptake CO2. Much like what happens with the ocean working as a buffer against climate change, it's actually the microalgae in there that are uptaking CO2. So in theory, if you put them in an industrial environment, they should help offset CO2 emissions. My study focuses on a pilot project. There's a basin full of water and microalgae in Palavas, and they have microalgae being cultivated there. The issue that's arisen for them is that suddenly the microalgae will die, so the culture will crash completely. So they're trying to answer the question, why is this happening? This is where I come in and where other researchers come in, and we're asking, what's causing this? Could it be viruses? To do this, we have taken many water samples over growing periods for the microalgae. The water samples are filtered, and then there's an extraction process that takes place to take out nucleic acids of viruses. We work to isolate these different groups of viruses from the water samples. So we're studying DNA viruses, we're studying RNA viruses, and we're studying what's known as giant viruses that are occurring in this basin. So giant viruses are, like I said, giant in terms of their physical size and also their genomic size. And because we take them over time, we can also investigate how the basin is changing over time. This is done by a laboratory technique called qPCR. Essentially, you make up an assay, so we can watch a virus appear, we can watch it multiply, we can watch it rise in its number, and then we can see if it plateaus, if it dies off, etc. And so we can take this information and coordinate it with when the crashes occur in the system to see if this interaction is occurring at the same time. In order to figure out what viruses are in the system, we have to use a metagenomics sequencing approach. So this is where computers come in to aid our understanding. When we do sequencing of these viral samples, there's a lot of data. So this involves coding, scripting, using Python, using R, using clusters, etc. to try to figure out which viruses are in there. So microalgae are single-celled organisms, and there's a lot of evolutionary questions that you can answer by looking at their interactions with viruses. What I get out of this project is learning more about evolution, but I'm also gaining some more tools to ask evolutionary questions, and that was one thing I was quite interested in.